RecBCD is a 3-subunit enzyme, as I've already said. And once it's entered a double-strand break, 2 of those subunits are motor proteins. The RecB protein, which is this one on the top strand, is a motor protein, helicase-like protein that moves on DNA in the 3' to 5' direction. The subunit has a nuclease activity, which can cleave DNA strands, and it will cleave the different DNA strands as it's directed to by a mechanism that's controlled by those Chi sites. The second motor protein is this D protein here, which is on the bottom strand. It is actually a faster motor than its other friend, the B subunit, and it moves only in the 5' to 3' direction. Okay, so here we have RecBCD moving along the DNA. The 2 motor proteins are helicases, so they break the hydrogen bonds in the double-stranded DNA. So we have a fork here, and it's moved along before it hits a Chi site. The nuclease reaction occurs to give rather frequent cuts of the 3' ending strand and rather rare cuts of the 5' ending strand. The frequency of the cuts really has a lot to do with the fact that the bottom strand has the fast motor and the top strand has the slow motor. So they're sharing the nuclease, so the nuclease has more time to cut the top strand than it does to cut the bottom strand. Now we have the enzyme encountering the Chi site, and that's where its activity is going to be changed by the fact that it's interacting with that DNA sequence. So the DNA sequence is recognized specifically by the RecC enzyme, and when RecC binds to that sequence, it causes a fundamental change in the whole protein complex. Basically what it does is it turns off the RecD motor protein, and that was the fast one on the bottom strand, which is the 5' ending strand. So now the only motor we have working on this enzyme is the top strand motor, the B protein, which is the slow one. So this is really critical for how this enzyme achieves making the structure that has a long single-stranded region that ends in a 3' end. So this is the 3' end here. This is the 5 to 3 strand. And after the Chi site, the enzyme only cleaves the bottom strand. It doesn't cleave the top strand anymore. And it's now cleaving the bottom strand more frequently because it's moving slowly. And this process generates a DNA molecule that has a 3' single-stranded overhang from a DNA that was once just a flush double-strand break.